Good morning, students. Our today's topic is photosynthesis in higher plants, that is, Chapter 13. All animals, including human beings, depend on plants for their food. Green plants, in fact, have to make or rather synthesize the food they need, and all other organisms depend on them for their needs. Green plants carry out photosynthesis, a physico-chemical process by which they use light energy to derive the synthesis of organic compound. So during photosynthesis, plants use inorganic compounds and form organic compounds like glucose. Ultimately, all living forms on earth depend on sunlight for energy. The use of energy from sunlight by plants during photosynthesis is the basis of life on earth. So, photosynthesis is very important. It performs two functions mainly. First one, it provides the food to all the organisms on the earth. So it is the primary source of food for all organisms. Second, photosynthesis is responsible for the release of oxygen into the atmosphere by green plants. So during the process of photosynthesis, green plants releases oxygen. So for oxygen and food, photosynthesis is required. Next is, what do we know? Chlorophyll, that is a green pigment of the leaf, light and carbon dioxide are required for the photosynthesis to occur. These are the things which are required for the process of photosynthesis. Now, few examples or few experiments. That starch formation occurs in the leaves. That is, first take two leaves, a variegated leaf or leaf that was partially covered with black paper. Variegated leaf means a leaf in which green or yellow colors are present, or take a leaf and cover that leaf with the black paper, some part of the leaf, and then expose that leaves to the sunlight with the plant. On testing these leaves for starch, it was clear that photosynthesis occurred only in the green parts of the leaf in the presence of light. When we place both the leaves in the sunlight with the plants, the part of the leaf which is covered with black paper, that will not perform the process of photosynthesis, and so in that starch formation will not be there, and that part will not show the starch test. But the other part which is green and perform the function of photosynthesis, that part show the starch test. Another experiment which will show that carbon dioxide is essential for the process of photosynthesis, that is the half leaf experiment, where a part of the leaf is enclosed in a test tube containing some KOH soaked cotton. So KOH has the property, KOH means potassium hydroxide, it has the property to absorb carbon dioxide. So when we set up this whole apparatus outside in the sunlight, on testing for starch later in the two halves of the leaf, we will find that the exposed part of the leaf tested positive for the starch test, while that the portion which is inside the tube tested negative. This showed that carbon dioxide was required for photosynthesis. The part which was inside the test tube, there the carbon dioxide which is produced, that is taken by the KOH solution. The part of the leaf which is inside the test tube will not perform the function of photosynthesis because carbon dioxide is required for photosynthesis. Early experiments. Joseph Priestley in 1770 performed a series of experiments that revealed the essential role of air in the growth of green plants. Priestley discovered oxygen in 1774. Priestley observed that a candle burning in a closed space, a bell jar, soon get extinguished. Similarly, a mouse would soon suffocate in a closed space. He concluded that a burning candle or an animal that breathed the air both somehow damage the air, because during respiration they give out carbon dioxide. But when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn, because when the plant is present inside the bell jar, during the process of photosynthesis, the plant will take carbon dioxide and give out oxygen, and that oxygen will be used by the candle for burning and by the mouse. Using a similar setup as the one used by Priestley, but by placing it once in the dark and once in the sunlight, Jan Ingeshouse showed that sunlight is essential to the plant process that somehow purifies the air fouled by burning candle or breathing animals. The burning of the candle or breathing of animals, they impure the air because carbon dioxide is released during this. Ingeshouse is in an elegant experiment with an aquatic plant showed that in bright sunlight, small bubbles were formed around the green parts, while in the dark they did not. Later, he identified these bubbles to be of oxygen, because during sunlight the plants perform photosynthesis. During photosynthesis, oxygen is released by the green part, so that will appear as bubbles on the plants near the plant. Hence, he showed that it is only the green part of the plant that could release oxygen. Julius von Sachs provided evidence for production of glucose when plants grow. Glucose is usually stored as starch. Plants store glucose as starch. Then T.W. Engelmann, using a prism, he split light into its spectral components and then illuminated a green alga, that is cladophora, placed in a suspension of aerobic bacteria. एंगलमैन ने वाइट लाइट को स्प्लिट किया, वो वाइट लाइट 7 कलर्स में स्प्लिट हो गई, दैट इज़ विबग्योर. उस लाइट को उसने ग्रीन अलगा क्लेडोफोरा पे डाला और उसमें आसपास में कुछ एरोबिक बैक्टीरिया भी प्रेजेंट थे. द बैक्टीरिया वर यूज्ड टू डिटेक्ट द साइट ऑफ ऑक्सीजन एवोल्यूशन. He observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. In the Vibgyor light, red light is present, blue light is present. So these bacteria are present in the blue and red region of the spectrum. A first action spectrum of photosynthesis was thus described. It resembles roughly the absorption spectra of chlorophyll A and B. The red and blue light shows that the chlorophyll A and chlorophyll B molecules also absorb this spectrum of light only. So he concluded that bacteria are present in this region because oxygen is released and these aerobic bacteria are present near the oxygen, that is near the red and blue light. The empirical equation representing the total process of photosynthesis for oxygen evolving organism was then understood as carbon dioxide plus water in the presence of light. It forms CH2O plus oxygen, where CH2O represents a carbohydrate. A milestone contribution to the understanding of photosynthesis was that made by a microbiologist Cornelius van Nijl, who based on his studies of purple and green bacteria demonstrated that photosynthesis is essentially a light dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates. So they express the reaction as H2OA plus carbon dioxide in the presence of light forms 2A plus CH2O plus water. In green plants, water is a hydrogen donor and is oxidized to oxygen. Some organisms do not release oxygen during photosynthesis. So when hydrogen sulfide is used instead of water, that hydrogen sulfide is the hydrogen donor for purple and green sulfur bacteria. The oxidation product is sulfur or sulfate depending on the organism and not oxygen. So when hydrogen sulfide is used, sulfur or sulfate comes out, and when water is used, then oxygen comes out. Hence, he inferred that oxygen evolved by the green plant comes from water and not from carbon dioxide. This was later proved by using radioisotopic techniques. The correct equation that would represent the overall process of photosynthesis is carbon dioxide plus water in the presence of light forms glucose. The formula is C6H12O6 plus water plus oxygen. The oxygen released is from water. This was proved using radioisotope techniques. Can you explain why 12 molecules of water is used as substrate? Answer is, when 12 molecules of water are used, from that 12 atoms are used for the formation of one molecule of glucose and 12 atoms are used for the formation of water molecule. So from the above reaction we can show that 12 atoms are used in glucose and 12 atoms are used for the formation of water. Next topic we will continue afterwards. Thank you.